Welcome and thank you all for joining us today. My name is Brielle and I'm one of the organizers for this year's Quantitative Seminar Series for the Converse Lab. And I'll begin by acknowledging the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. We also acknowledge that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So today I'm very excited to introduce our speaker today, Beth Phillips. Beth is a research fish biologist at NOAA Fisheries Northwest Fisheries Science Center. She works on the Fisheries Engineering and Acoustic Technologies team in the FRAM division. She completed her PhD at the University of Washington in SAFS in 2018 and holds a master's in marine biology from Moss Landing Marine Laboratories as a part of San Francisco State University, and a bachelor's in biology from Western Washington University. Her current research is focused on investigations between dynamic physical ocean conditions and multiple trophic levels, including zooplankton, fish, seabirds, and marine mammals. So thanks again, Beth, for agreeing to give a talk today, and we're looking forward to it. So thank you.
All right, thank you everyone for joining. I'll just say up front, I cannot see anybody or anything besides my slides, which are to my left, and my camera's here, so forgive me when I'm looking in different places. Okay, so let's see if this works now. Okay, so again, thank you, Brielle, for that nice introduction and for everyone for being here. I am a member of the Fisheries Engineering and Acoustic Technologies, or FEAT team at the Northwest Fisheries Science Center. And so for today's talk, I'm going to cover sort of a very brief history of sound in the sea, and then get into some of the important acoustic sampling principles that we use for our fisheries acoustic surveys at NOAA. Then I'll describe how the FEAT team uses acoustics to conduct our Acoustic-Trawl survey for Pacific Hake along the west coast of the US and Canada. And then finally, I'll describe some upcoming research that we are conducting that's taking advantage of recent advances in echo sounder capabilities. So the fact that sound can be heard in the sea has been noted for a long time. Aristotle was one of the first actually to note that sound could be heard in water as well as in the air. And then nearly 2,000 years later, Leonardo da Vinci made the observation, shown as the quote on the slide, that ships can be heard at great distances underwater. In 1687, Sir Isaac Newton published the first mathematical theory on how sound travels. And while he was focused on sound travel in air, the same basic theories apply to sound in water. So the first successful measurements of the speed of sound in water were made by scientists in 1826, who recorded how fast the sound of a submerged bell traveled through water in Lake Geneva in Switzerland. And they estimated that the sound traveled at around 1,435 meters per second in fresh water, which turned out to be quite accurate. And it's around this time in the 1800s that scientists began to think about practical applications of underwater sound. And I'm going to skip through a lot of advancements in the study of sound in water that don't apply specifically to fisheries, but suffice it to say that a lot of exploration, including a lot of defense, thinking about sonar and submarines, relies on the theory of how sound travels through water. So the first successful experiments on using acoustics to detect fish were reported by scientists in 1929 from Japan. And they had noted these sort of unexpected signals in the mid-water column that they attributed to fish schools. Further advances came throughout the 1930s and 40s with echo sounders that were able to record data and produce echograms on paper, as shown here in the picture in the middle of the slide. And from the 1930s onward, scientists focused a lot of experiments on detecting fish and characterizing fish schools using echo sounders. Since then, from the 60s all the way through, basically through the 90s and through present times, The power and the resolution of sonars has continued to improve as new instruments were designed for fish detection. And many of these are used in fisheries research. And a lot of the characteristics that have improved include digital signal processing and displays of multicolored echograms, the use of split beams and multiple frequencies on echo sounders, as well as multi-beams, and more recently broadband signals that I'll touch on at the very end of this talk. So what exactly is sound? Sound is created by a vibrating object, and it's a pressure wave that travels through a medium such as water. A sound wave, therefore, is a longitudinal wave, and this is an animation that I'll show you of a sound wave's propagation, and it's traveling at a rate of speed that's defined by c, which is the speed of sound. And it's typically expressed in meters per second. You can see the amplitude of the wave varies from low to high. I don't think I can show with my cursor, but you can see the figure in the middle there. The amplitude is varying from low to high pressure. And this is where the particles, in this case water particles, are being either compressed, as shown in the darker blue regions in the middle figure, or expanded, representing areas with lower intensity or lower amplitudes. So this is, let's see, it should be noted that the sound travels as a wave, but the individual particles that are getting displaced don't actually travel, don't move. They only vibrate back and forth as the sound wave moves through the medium. So let's see here, and then, and then we can say that I wanted to point out that the wavelength is the distance between the peaks in amplitude of a sound wave. And then you can see again the high pressure and low pressure regions on the graph. And another thing I wanted to note is that the frequency, which is often reported in fisheries acoustics in hertz or kilohertz, relates to how often there are these peaks in amplitude or intensity, and it's a function of speed of sound divided by the wavelength. Okay, so this is an example of a single frequency transmitted pulse, and this is at 38 kilohertz in this example. And you can see it's a constant amplitude, so the peaks and the intensity are always the same. And it's a transmitted pulse. The transmitted pulse duration is the amount of time that it's transmitted. And this is typically referred to as a ping. And you'll hear me refer to that later on too. And you can also see on the right-hand figure, there's a plot of a pulse spectrum of the sound wave, and you can see its greatest intensity again is centered right around 38 kilohertz in this case with this example. So how is sound actually, so that's sort of the fundamentals of sound waves, but how is sound actually transmitted into the ocean? So transducers are instruments that generate acoustic pulses from oscillations of piezoelectric elements, which is shown here as the orange box. And a transducer is made up of many of these elements that together each emit pulses of sound, which travel outward from the transducer face. And as each sound wave travels outward and crosses over the path of another one, the displacement at that point is equal to the sum of the displacements of each individual wave. And this is essentially how a beam of sound is formed and transmitted through the water column. And you can see that there's both main lobes and side lobes. And what we really focus on is where the sound is at the greatest intensity, which is on the main lobe, which is directly, in this case, facing down from the face of the transducer. You may have heard fisheries acousticians refer to both near field and far field ranges of a transducer, and this is a really important concept because a target's scattering characteristics are different if they are in the near field or the far field. So within the near field range, the wave fronts, as you can see in the figure on the lower left, produced by the transducer are not yet parallel, and the echoes from targets within this region can vary greatly with small changes in the location of the target in the sound beam. So therefore, we focus all of our efforts primarily on analyzing targets that are detected in the far field range, and that's where the wave fronts have become very well organized and the echoes from targets are more predictable. And you can kind of see that in the top graph there with the far field range, and it also aligning with the main lobe of the sound beam, which is the greatest intensity. Okay, so now I'll just cover in very broad terms the sonar equation, which is a really important, basically the way that we interpret all of the backscatter that we record on our echo sounders. And then we use that to estimate fish abundance and biomass. So the sonar equation includes components of energy that are sent out from the transducer, which is the sort of this basic equation of E sent, and it's minus energy lost during transmission. And then it also includes energy that's reflected back from a target, minus any of the energy that's lost during the reflection of that sound back to the transducer. And so all of that put together is what is received at the echo sounders at the face, and what's recorded is the energy that's received. So there's sort of two components that I mentioned. The transmission loss is just both energy lost out and lost back, and then the energy that's reflected from a target. And that's what's really of interest to us when we're studying fish in the water column using acoustics, is that energy that's reflected from a specific target, and that's what we're trying to quantify. So as far as the transmission loss, there's two components I wanted to touch on. So the sound intensity at any given point decreases with increasing range from the transducer, from the source of that sound. And there are two components: spherical spreading, which is shown in the figure as the beam spreads across a larger volume as it moves down through the water column. It spreads outwards. And so the intensity is basically spread over a larger and larger volume. So that's termed spherical spreading. And the second component of transmission loss is absorption. And that's the energy that's just lost simply to the friction and vibration of those water particles as the sound wave is passing through it. So these are both very important components to keep in mind when we're trying to think about detecting fish or any other targets in the water. And I also wanted to note that this transmission loss is a sort of two-way process. So there's some loss in the intensity of sound as it's transmitting out from the vessel or from the transducer, and then also as it's being transmitted back and getting recorded. Okay, so that's the very basics of sound transmission and the sonar equation. But what we're really interested in, as I mentioned, are the actual targets that we're trying to detect that are scattering sound back to the fisheries research vessel or the echo sounder. So this is a figure showing fish being detected by a transducer, and each target in the beam has a target strength, and that's the amount of energy that it reflects when a sound wave hits it. And every single target has a specific target strength. In this case, each fish is sufficiently separated from each other in the sound beam that individual targets can be measured. And that's typically referred to as either target or echo counting. And this is really sort of the origins of how fisheries surveys were first conducted, where they were able to detect individual targets and begin thinking about how to count those individual targets, or in this case, fish. However, we know that in most of the areas that I work, at least in most of the areas of the ocean, we study fish that form aggregations, either large aggregations of near the seafloor or in the midwater, or schools of fish. And so it can make it very difficult to detect single targets. So instead, another way to think about quantifying fish acoustically is to consider what's termed the volume backscattering strength, or SV, which represents the summed contributions or the target strengths of all of the individual fish that are detected, and then that gets normalized to a sampling volume. So this is effective when there's more than one target within a close distance to each other in the beam, either horizontally or vertically. So you can start thinking about like aggregations of sardines or herring where they're really tight balls and you really can't discriminate individual fish. This is the approach that we use to quantify the backscatter from those targets. So I also wanted to mention that SV is a really valuable measure of backscatter because it can be used to estimate the abundance of fish in an aggregation or a school through a method called eco-integration. And this is where volume backscattering strength is integrated across an area to produce an areal density estimate, which is you've probably heard of referred to as NASC, which is the nautical area scattering coefficient. A lot of fisheries acousticians refer to this term as NASC. And this is a density measurement of fish for a column of water with a certain surface area of 1 nautical mile squared, and then a specific height, which can be the full depth of the water column or a specific height of interest. Let's say, mid-water region that you're interested in studying. Okay, so all of this is just to hopefully try to help you understand the echograms, which is shown in this slide here, are images of backscatter collected by an echo sounder during a fishery survey. Basically, they're not just pretty pictures, although I think they are quite pretty. But they actually contain a whole lot of data. And in this image, you can see this is an example of Atlantic mackerel detected using an echo sounder operating at 200 kilohertz. You can see the depth on the y-axis from the sea surface down to about 60 meters. And you can see these really large aggregations of fish across a distance as the vessel was moving along the surface of the water. You'll also note that the colors here, the bright reds and the hot pinks, are actually, if you look on the right on the legend there, are actually representing higher intensity backscatter, and it's shown here as SV, which is in decibels. And so the pinks and the reds are stronger SV values or higher SV values, indicating more fish in that volume of water. So now I wanted to just walk you through to demonstrate that the echograms have a whole bunch of data that's so useful. So we can start zooming into one small aggregation or ball of Atlantic mackerel in this case. And you can start seeing, as you begin zooming in, you still see the colors, and the SV values are represented on the right. There's some high values of pink, there's some greens, there's some yellows. But you can start seeing, if you look vertically from the top, I don't think you can see my cursor, but from, let's say, the 440 meter cell bin and distance on the top, and just look straight down, that sort of column is one ping that was transmitted by a transducer as the ship was traveling by. And you can see within that one ping all of these different values of SV. And those are all different samples that the transducer recorded as the sound was being transmitted back up to the sea surface. And let's see, here we go. Okay, so now we're actually starting to think about measuring that SV in each of these individual cells within each of these echograms. And you can see that there is a very specific SV value attached to each of these cells. And then we're going to keep zooming in. And here you can see I've highlighted SV value in one of the cells that's minus 57.0433 decibels. And this can actually be integrated across that area, again, using echo integration to produce an estimate of fish density. And then that density can then get measured and multiplied by the area to get the actual abundance or numbers of fish. Okay, so all of that is dependent on two important things. One is that your SV, or volume backscattering strength measurements, are truly accurate and correct. And that relies heavily on making sure that your echo sounder has been calibrated properly, so that all of the data that you are sending out, all of the sound waves that you're generating and the backscatter that you're recording are correctly transmitted. The location and the sound beam and the beam widths and all of that have been corrected, so that you know that what you're receiving back is actually correct. And it also relies on knowledge of the target strength that is generating that backscatter. And so going back to what I talked about earlier, every single target that's detected acoustically has a specific target strength. And so if you know that information, then you can start thinking about integrating the SV, and then thinking about individual targets, let's say of mackerel in our example, what their target strength is for each fish, and then dividing that by the total amount of fish that were in that cell to estimate, to get from the volume backscattering, which is again the whole aggregation, to get to actual individual numbers of fish. Okay. Oh, and I just wanted to note that I mentioned calibration. I'm not going to talk about it today, but it is a very critical component, especially for scientific fisheries surveys. Okay, so as I said, target strength is a measure of the energy backscattered by a single object, in this case, a fish. And it's generally proportional to fish length, which is an important element to keep in mind too. And fish with swim bladders tend to reflect sound strongly at frequencies around 38 kilohertz, which is oftentimes why we use that frequency for our surveys. And this can be modeled or directly estimated. And so there have been many different ways to estimate target strength of different targets of interest, whether it's mackerel or herring or hake. The first one is just theoretical scattering models, where there's no fish involved, but they take into account the length of the fish, the radius of the swim bladder, in particular if it's a fish with a swim bladder, as well as angles, the frequency at which the fish produces the strongest sound, and they can then sort of estimate, like theoretically, what the target strength would be. Other research has used both live fish in cages as well as fish that have been tethered, and used echo sounders to then record the backscatter, and then, with knowledge of this length of the fish, the size of the swim bladder, other parts that might be scattering sound, can then estimate target strength. And then finally, the last example here is estimating target strength from wild fish, again, provided that the targets are known and can be ground truthed or captured and identified, and their lengths measured. So, what just happened? Did my slides go from?
Yes, now we see the PowerPoint again.
Okay, so let me go. That's strange. Let's do this. And then let's go to there. Okay, oh. Something is happening here.
That looks good.
Is that working? Okay. I don't know, it was turning blue for a second there.
Oh, now it's blue. Oh no. But now it's good. Now it's good. Okay. It was blue for a second. But.
Okay, don't move. Don't touch me. Okay. So there's okay, so now my slides are messed up again. Okay, so there's a number of positive aspects of using fisheries acoustics in fisheries surveys, including the fact that sound can be used to sample a very large volume of water, and also provide really high spatial resolution due to the number of pings that can be transmitted and received per second, and then of course as the ship moves along at speed. Acoustics can also sample a wide range of targets from small zooplankton all the way up to very large pelagic fish. And it's a remote sensing technique, so it's non-invasive. It essentially offers a way to survey fish without actually removing them from the ocean. There are, however, some cons. Again, as I just explained, it requires knowledge of the target of interest and the target strength of the fish you're trying to study. And then it oftentimes requires additional ground truthing to verify species identification and, of course, obtain additional information on lengths and ages and everything else that you can't get using acoustics. This is in comparison to direct sampling, the more sort of traditional surveys for fish using either nets or traps, hooks, photos or videos, which are obviously by nature extractive and or at least invasive. Most of the methods that we use for fisheries surveys remove the fish and kill them. Depending on the sampling method, the size of the mesh of the net, the turbidity in the water if you're using videos or photos can really limit the amount of area or volume that you're sampling. There's also relatively poor vertical and horizontal resolution depending on how fast the ship can move while you're trying to trawl. So in comparison to especially fisheries acoustics, it's typically a much smaller area that's sampled. And of course, we know there's a lot of size selectivity, especially with certain nets and the size of the mesh that can limit how much information we can glean from direct samples. But as I mentioned, there are some positives too. We can obtain information on age and length, other biological samples, including information on the sex of the fish, diets, DNA, etc. And in particular, it can be used to ground truth acoustics. And so many scientific groups have taken advantage of sort of both of these approaches and sought to sort of integrate them into what's termed an acoustic-trawl survey. And this is what my team, the FEAT team that I work on, has been doing for quite a while. And so we conducted an integrated acoustic-trawl survey along the West Coast for Pacific Hake. So now I'm going to describe some of how we're applying this acoustics and trawl methods to a specific case study here with hake. So hake is an international fishery. It's fished by the US and Canada along the West Coast throughout the spring and summer and into the fall. It's the largest single species fishery by volume on the US West Coast. And that represents a landed value of over $45 million with an economic impact to the whole industry of approximately $200 million. So it's a quite valuable fishery. Hake are also ecologically important, both as prey and predator. They are prey for a number of birds and mammals and fish, as well as each other. They are cannibalistic, but they're also important predators on other important components of the food web of the California current, including krill and squid, other fish like herring, and also each other. Another important aspect of the Pacific hake population is that the biomass can fluctuate substantially over space and time. And this map on the right is showing a picture of sort of their general migration. So hake are thought to spawn during winter off of Southern California, shown as the gray area on the map, and then migrate north along the coast in spring and summer to feed. And they form these large feeding aggregations along the coast. The proportion of adult hake that actually migrate and move into Canadian waters can vary each year, and it's been tied to the strength of the California undercurrent as well as temperature, and there's a lot of variables there. Another thing about hake is that they can live to be approximately 20 years old, and so large changes in their biomass are often driven by shifts in the dominant year class as each year class moves through the different age classes. And so because of that, and because it's such a valuable fishery, it is a really important species to continue monitoring, and that's what our work has been focused on within the FEAT team. So we operate the Joint Integrated Acoustic-Trawl Survey, which provides a fisheries independent set of data on the biomass of hake for the use in stock assessment. So we specifically estimate total biomass of hake and the composition of adult hake on the west coast of US and Canada. And you can see the map on the right here shows our survey area, which extends from Point Conception in Southern California to Dixon Entrance in Southeast Alaska. And we conduct line transects that run from east to west, from the 50-meter isobath all the way out to 1,500 meters. And we begin in the south, and the location of the first transect is sort of randomly chosen, and then we proceed from there. Oh, it happened again. I'm going to try to just see if I can get back here again. Let's see here. I'll do this. Oh, no, this. Okay, are we back?
That looks good.
Okay, I don't know why my computer keeps doing this.
Now it's good.
Okay, hopefully we can keep going. Okay, so let me catch up here. The transects are 10 nautical miles spaced apart, moving up the coast. We survey in summertime, biennially, and it typically takes about 90 days total to complete our survey. We operate during the day, conducting our acoustic sampling and trawling to ground truth our acoustics. And then we also operate at night, collecting a lot of oceanographic data. Okay, let's see if this keeps working. Hopefully it does. So this is an example of an echogram showing sort of a cross transect slice of what we see on the 38 kilohertz echo sounder as we move along our transect. The seafloor has been blacked out here, and the echogram you can see is showing a strong backscatter represented by the reds and yellows and oranges between 300 and 400 meters water depth. So this is what we do most of our time. What most of our time is spent doing is scrutinizing these echograms and determining whether or not we need to put a net in the water and sample it to determine if it's indeed hake, and if it is, obtain additional information about the aggregation in terms of the length, the sex of the fish, and all the other biological data I mentioned. We typically conduct between 2 and 3 trawls per day, and it's always, it's a continuous trade-off between covering our whole survey area and then ground truthing our acoustics. We typically conduct about 100 trawls per survey total. So this is just now an example of what we get when we put our net in the water. You can see these pictures of the trawl net. It's an Aleutian wing trawl. It's a mid-water. We try to sample, aggregations as best we can that we suspect are hake. And then we get a nice, hopefully, haul of hake, and we process them, and we get this plot on the bottom showing the length frequency distribution of the trawl catch itself. From that, then we can start looking at our echogram and classifying different regions as either hake or not, depending on our catches. So this is sort of the process. Just quickly step you through it. We identify hake aggregations acoustically. We confirm them with trawling. We then define hake regions on each of the echograms based on the characteristics of the acoustic backscatter and the trawls that we've conducted. And this is now a plot showing for 2019 the total length distribution of all the fish that we caught across both the US in blue and then in Canada as well. And you can see our line transects there. So from this, we have an analyst defined stratification process that I'll show you in a moment. But then you can see from that stratification process, we can start thinking about looking at hake distribution, which is, again, using echo integration from our acoustic data from the backscatter to estimate, again, NASC, which is the areal density. And now on the map on the right, what you're seeing in those red circles is the estimated NASC attributed to hake for our survey in 2019. And you can see that, for this year, there was a lot of hake off of Cape Mendocino and Northern California and Central Oregon, fewer up in Canada. And then, from NASC, I'll show you in a moment, we can start thinking about estimating, again, total hake numbers, and then moving towards estimating hake biomass from the total numbers. And I just wanted to point out that we are focused on sort of separating age 1 hake, which are the young of the fish that aren't part of the fishery, but can be used as an index of recruitment, from what we term adult hake, which are age 2+ hake, that are the assessed stock. So this is an example of taking the length frequencies from our trawls and partitioning them into different strata. And we use Kolmogorov-Smirnov tests to define the strata based on the distributions of the lengths and the locations and the characteristics of each individual hull. We do sort of pairwise comparisons then to classify each different strata. So I'll just walk you through here. You can see, so strata 1 were actually age 1 fish that were less than about 30 millimeters in length. So those were excluded from our estimate of the adult hake biomass. But the rest, now you can see, are different distributions of fish based both on their length frequencies and then also the locations and characteristics of each individual hull. And then we sort of stratify each of these groups. And in this case, we came up with five strata. From this information, then we can start partitioning the backscatter along all of the transects into these different strata, and then estimating target strengths based on the length of these fish, and then estimating abundance. So there we go. See if this is going to work. There, okay. So this is just another example of sort of walking you through the process here. So we have our acoustic echogram, we have our region of hake that's been identified, we have the trawl information for the lengths that were in this aggregation. We can use echo integration to estimate the nautical area scattering coefficient and the volume backscattering strength within this aggregation, within each of those cells on our echogram. And then because we know hake length, we can use the established hake target strength length relationship to then estimate target strength of hake, and then divide the total backscatter that we got from this region in our echogram by the known backscatter from one individual fish. And so walking through this step-by-step equation, we will end up with the estimated number of fish in that aggregation. That's a very simplified version of it, but hopefully it makes sense. And then from there, once we have the numbers of fish, we can then just turn to our trawl data, where we have both length and we have the weights of these fish. We can use a length weight regression to then estimate the weight of fish at a given length. And then from there, we can estimate the mean weight of fish at any individual length by the total number of fish in that length class. Okay, so that's how we get to biomass along all of our transects for hake. That was, again, detected acoustically, classified, and then we get to the biomass. But then we want to sort of estimate total biomass in our full survey area. So for that, we use kriging, which is an interpolation method that relies on the weights of neighboring points and takes into account sort of the underlying spatial correlations within the data itself. And so this figure on the right is showing an echogram for hake biomass along our transects. And it's measuring, the variogram is basically a measure of how variable things are as distance increases. So on the x-axis here, you can see distance in nautical miles. And on the y-axis, you can see the correlation coefficient. So the red line here is the average correlation. We do look both, thinking about our transects are oriented east to west. We think about looking at correlations both along isobaths, so moving up and down the coast along like the 500-meter isobath, for example, but then also across. And you can see there's different correlations there. We take the average of both of those to look at the variogram, and then use that as a input to do the actual kriging. And it's nice because this also generates an estimation of the error in our data. And so we can get an estimate of the error around our biomass estimate, but also see how well the model is performing. And what you can see here also, I wanted to point out that, again, our transects are spaced 10 nautical miles apart. You can see the correlation drops to below around 0.3 at about 5 nautical miles, so sort of within the distance between each of our transects. Okay, so now let's see if we'll open this again. There we go. Oh, there we go. Okay, so now this is just sort of the final product that we get from that kriging exercise. So we have our input data as the biomass along the transects, and then we use kriging to then estimate biomass onto a grid that the cells are 2.5 by 2.5 nautical mile cells that we can grid, that we can krige and estimate biomass across the entire survey area. And so that's what's shown here for 2019 in the map in the middle. And then again, we also get an estimate of the coefficient of variance for the kriged data. And you can see it's actually quite low between each of our transects. On average, it's around between 4 to 6%. So it's a relatively precise measure. So then that is what ultimately ends up getting shared with the stock assessment team. So on the left here is a figure of our biomass estimate from the acoustic survey. And you can see it's a time series from 1995 all the way through, we actually have it through 2021, but this shows through 2019. And then that is then used by the stock assessment team at NOAA to produce their estimate of the total relative spawning biomass of hake as one of their inputs into their model. And for more information, if you're interested in the actual stock assessment itself and status of hake, you can see there's a website down here you can go to that has all of that information. Okay, so I've covered a lot of stuff here today, and primarily focused on research that we do related to Pacific Hake, and a lot of it relies on advances in technology that have occurred over the past 30 to 40 years, as I mentioned earlier. Split beam echo sounders using multiple frequencies, and then now what's happening is we're moving into using more broadband and broadband split beam echo sounders, and that's sort of where we are with present, and then moving potentially into the future with multi-beam broadband. So I haven't really told you much about the differences, what broadband even is, but what I've been really kind of focused on in this talk, and what we focus on with our research, is primarily with the fisheries survey, is using narrowband signals from echo centers, and in most cases we use 5 different frequencies, and that's what's shown here in the top. So they're sort of the greatest intensity is centered right around each of those frequencies. I showed you earlier the 38 kilohertz, but it can be the 120 or whatever it is. And that's where we can base a lot of our information on target strength of different fish, where their greatest intensity of the backscatter produced by, let's say, a hake is at 38 kilohertz. So we can focus on measuring data collected at 38 kilohertz. But broadband has been more recently developed, and it can actually transmit across, as it implies, across a wider range of frequencies. And there have been new equipment that's come online, new echo sounder systems that can transmit and receive broadband signals across this sort of a wider range of frequencies, which improves our ability to obtain information on spectral characteristics around the peak frequency of some of the scattering targets of fish, for example. And it also increases our range resolution vertically in the water column. And I'll show you an example of that in a second. So we are now at NOAA using EK80, Simrad EK80 system that can operate in both narrowband and broadband mode. And it can provide, therefore, information on acoustic signatures of targets over a much broader and continuous frequency band. And we just started using this last year in the 2020 survey. So it's really new and very exciting for us to start thinking about new research applications. The Simrad EK80 can actually transmit in both narrowband and broadband data. So we can actually collect data in narrowband, which is our typical survey that we will still use, to identify aggregations, obtain information on volume backscattering strength. But then we can switch to collect broadband data, which will, again, allow us to obtain additional spectral information that might allow us to think about improving our ability to classify different species. So comparing hake versus, say, rockfish or other fish that might be scattering in the water column around where we're trying to identify hake aggregations. We can also start thinking about improved resolution, particularly when we're trying to discriminate hake or other aggregations of fish really close to the sea floor, which oftentimes is more difficult because the backscatter produced by a hard surface like the sea floor can oftentimes sort of hide a lot of the sound scattering from fish. And then we can also start thinking about estimating size, for example, of zooplankton like krill, based on their scattering properties that are recorded across a wider frequency range. And of course, with all of this work, we are still going to have to pair it with direct sampling to ground truth a lot of the acoustic data. But both the Bell M. Shimada, which is the NOAA research vessel that we operate for our hake survey on the West Coast and US waters, and the Sir John Franklin, which is the Department of Fisheries and Oceans Canada vessel that they use in Canadian waters, have these systems, as well as a number of other new acoustic systems that we're going to start using this summer to start investigating some of these questions around acoustic scattering and classification. So with that, I hope that I've demonstrated how we use fisheries acoustics, at least in the Pacific Hake stock assessment, to estimate biomass and provide that for the stock assessment team. And maybe impart to you the value that acoustics can bring to fisheries research. And with that, I wanted to just acknowledge both scientists at Fisheries and Oceans Canada, particularly Stéphane Gauthier and Chelsea Stanley, as well as the team at the Northwest Fisheries Science Center. In particular, Sandy and Julia have been fabulous. And I also wanted to note both Rebecca Thomas and Desang Chu, who have helped me learn a lot of this stuff in the last year and a half or so that I've been with them. And they are far smarter than I am and can probably answer all of these questions much more elegantly than I can. And Chu in particular, he has been with this group for 20 plus years, and he's actually going to be retiring this year, and so he will be greatly missed, and he's contributed a lot of research that I didn't even touch on to fisheries acoustics and both our team and international work on acoustics. So with that, I would like to thank you, and I may have time for questions.
Yeah, thank you so much for giving that introduction. I learned a lot, kind of took me back to physics days and thinking about waves and all of that, so that was cool. Yeah, as a reminder for everyone, you can put questions into the chat and I will read them out loud. And also, if you prefer to just voice your questions, I can unmute you if you would like to raise your hand. Yeah. So our first question is from Jen Zaman, and Jen asks, will broadband help with resolving fish or targets that don't have gas bladders, such as, oh gosh, I'm not a fish person, I'm sorry, euglin and sandglass and krill?
Oh, okay, yeah, so Eulicon.
Yeah.
Yes. Jen, yes. Hi, Jen. Thanks for tuning in. Yes, hopefully it will. Yeah, that's one of the things I think we're going to be exploring, not necessarily with Eulicon or sandlance, but certainly with euphausids for discriminating the two different species of krill that are common on the coast, Euphausia pacifica and Thycenoessa spinifera. They may have, with the broadband system, we may be able to detect slightly different peaks in their residence frequency that we might be able to use to discriminate species. So yeah, that's for sure something that I think could be applied to other species that don't necessarily have swim bladders either.
Cool. Jen says, hi, and thank you. So we have a question from Dylan Gomes. I'm sorry if I'm pronouncing your last name wrong. But Dylan asks, Dylan says, great talk, Beth. Can you speak to the behavior of hake moving during the surveys? You mentioned that the surveys take about 90 days. Do you have to take account for fish movement so that you don't double count or miss any?
Yeah, that's a great question, Dylan. Thank you. So yeah, our assumption when we're doing our surveys that the hake have essentially stopped their migration and have found an ideal habitat to be foraging, but that is an assumption. And so there is the potential that some fish may be continuing to move north, or even have decided to move back south again. And we've been exploring that question in a few different ways. One of the main tools that we've used that I didn't mention at all today is sail drones, which are uncrewed systems that are basically drones that use wind to sail along, and they can be programmed to sail along different transects with waypoints. And they are equipped with echo sounders as well. And so this last year, we actually deployed 2 sail drones in an area where we were suspicious of either hake continuing to be moving either south or north, or maybe even further offshore that we might be missing off of Cape Mendocino. And the sail drones essentially just like drove back and forth in the same location, right? So we're doing a transect and we're moving north and we're moving north and moving north. The sail drones just kept going back and forth in the same spot to try to answer that question of whether there are more hake moving through that we're missing. And so I guess the answer, I don't have the final answer, but it's something that we're definitely exploring. And, you know, there might be shifts in, you know, I guess stationarity, especially, you know, with changes in ocean conditions, potentially with maybe climate change, that we are definitely thinking about and are sort of at the front of our minds. Yeah.
Cool, thanks. So Karen Barcelo asks a question. Karen says, hi Beth, nice talk. Do you know if there are any acoustic surveys along Mexico that also discriminate hake?
Karen. So the Southwest Fisheries Science Center conducts surveys for coastal pelagic species, so anchovy, sardine, mackerel, and I believe they collaborate with scientists in Mexico to conduct surveys in Mexican waters for CPS. I am not sure if they are collecting data and looking at distributions of hake, but it's also something that we are talking about. Yeah. The assumption, of course, is that these hake most of them are down in the south off Southern California in the winter, spawning somewhere in the area and then moving north. But yeah, there's a lot of questions about like, is there another population of hake off Mexico and Mexican waters? How many are there? Are they part of the same population that we're doing our assessment on? Are they there year round, etc.? Yeah, so I'm not aware of any offhand, but I think I could talk to some people on my team. I'm sure they probably know a lot more about it than I do.
Cool, thanks. So there's a question from Yumi Aramitsu, and Yumi says, thanks for the informative talk, Beth. Because the EK80 system needs to be operated in either broadband or single frequency mode, i.e. it's not possible to collect data in both modes at the same time, how do you anticipate incorporating broadband information into more traditional acoustic trawl surveys in the future? And will surveys transition to broadband mode as the technology and analysis methods advance?
Oh, hi Yumi, thanks. That's a great question. So what we're doing, so our survey I mentioned, we conduct the full survey to estimate the biomass of hake every other year. And then in the off years, we actually continue to go out on the water, but we switch to do it focusing on more research. And so that is what we'll be doing this coming summer. And we will be sort of working with the EK80 system, collecting data in narrowband, but then switching when we can. Let's say we see some aggregations of hake that are near the seafloor, or we see some aggregations of hake that we might think are mixed with rockfish. We're going to be switching and collecting data in broadband mode after. Like, I think the idea is like we're going to run a transect with narrowband, collect all the data we can, turn around and go back over that same aggregation so we know it's the same, hopefully the same fish, and then collect data in broadband and then be able to hopefully compare that. So that's one thing. And then also we're going to pair that with our trawl nets, and we have stereo cameras too that we're going to put in the trawl nets to record the species composition of fish in the aggregation. So yeah, at this moment, the idea is that a lot of the broadband data from the EK80 is going to be sort of applied to various research questions that in the future we might be able to apply to our survey. For example, discriminating rockfish from hake acoustically without necessarily having to trawl on them all the time. Another thing we're doing, we have a project where we're working with folks at UW Applied Physics Lab to start using machine learning and artificial intelligence to process a lot of this acoustic backscatter, to start sort of automating that classification process. And I think that's where potentially a lot of the broadband data could be put in there. And we'll see how it performs, of course, and match it up to what we have from our survey. But I think there's a lot more research that needs to be going into stuff before we start putting any of the results into our survey. And then as far as switching to collecting data in broadband for our actual survey, I think I'm not by any means an expert, but I think that's pretty far ways off still, because a lot of the work so far has been done around target strength and everything else related to the narrowband systems. But again, that's an active area of discussion, I think.
Cool. So we have one final question from Alan Hicks. Alan says, thank you for the talk and great information. So when collecting data with broadband, are the data with a single frequency, like a 38 kilohertz, available as well to determine the quantities needed to calculate an index of abundance for a species, as has been done traditionally? Mm-hmm. Or do you have to switch between single frequency or broadband?
Yeah, so that gets kind of back to Yumi's question. So the broadband, it does, like, let's say the 38 kilohertz. So the broadband collects data right at the 38 kilohertz, but it also collects data sort of, I can't remember the range, but it's like 26 to 50 kilohertz or something like that. So it's this wider range. And so I do not know, this is a question for Chu, Alan, if we could use that broadband data right at that 38 kilohertz peak to estimate target strength and hake target strength and then biomass from that. I'm not sure exactly how that would work, but as far as I know, we're currently sort of still alternating between collecting data in narrowband and broadband, because I don't think there's been a lot of research on that yet. But I'll ask you.
Cool. Alan says thanks. Yeah, so with all of that, thank you again, Beth, for giving a talk today. It was really great. Yeah, learned a lot, and thanks everyone for coming up with the awesome questions too. And I will see you all next quarter. We have some great speakers lined up, so hopefully we'll see you all again then. Thanks again, Beth.
Thank you. Bye.
Thanks. Bye.